everybody, welcome to my talk. My talk today is going to be in the form of a conversation between me and my dad. Balaji, what are you doing in New Zealand for the last 3 years? Dad, I'm doing a PhD on drug-resistant influenza neuraminidase mutations. PhD on what? Okay, Dad, in simple words, let us call it PhD on influenza war. Have you heard about the influenza virus, Dad? Oh yes, it is the famous virus which caused swine flu in the year 2009. In fact, this virus was so famous that it appeared on the newspapers more often than Barack Obama did. I also remember Tamiflu, the drug which was used to treat swine flu. But isn't Tamiflu a failure? Not really, Dad. The drug was targeted against a specific viral protein called neuraminidase. The virus changed a single amino acid in its protein sequence. In biology, this process is called mutation. Now, because of the mutation, the affinity between the protein and the drug decreased. Hence, in some cases, the drug was not that effective. This is called as drug resistance. I see. So, you are trying to discover new effective drugs. Wow, my son is going to be a famous scientist. Well, thank you, Dad. But before investing millions of dollars in drug discovery, one important question has to be answered. Will the virus undergo any further mutation that is capable of causing drug resistance? This is the main objective of my PhD. I started my PhD with simulation analysis, studying the interaction between the protein and the drug. These interactions were measured with energy values. More negative the energy, more stronger the interaction is. The shocking news is that there could be more mutations causing drug resistance. But before I tell this to the world, I need to validate these results with experimental data, for which I'm going to use an SPR. SPR, what is that? That SPR is surface plasmon resonance. It is a biosensor used to measure the binding of biomolecules to surface-bound drugs. In this case, binding of the viral proteins to the surface-bound drug. Now, stronger is this interaction, higher is the SPR signal. Now, ideally, in a single experiment, I can study 36 such interactions. Really? 36 interactions in one go, and you still haven't finished PhD. Oh, come on, Dad. I've spent a lot of time using genetic engineering methods to make these viral proteins in the lab. I've also found out a way in which the drug can be attached to the SPR chip. I will now study their interactions in SPR. I'm expecting an SPR outcome where the normal protein could have a stronger interaction with the drug when compared to the drug-resistant proteins. I will use this data to validate my simulation results, with which I should proceed to the completion of my PhD. Now, Dad, please, enough of questions. If you want to know more about my thesis, please come to the Biointeractions Laboratory. Just remember that it is a PC2 lab, so please kindly follow the rules. Thank you very much.